This is Note Taker 14 on genetic engineering. Today we're going to start talking about what genetic engineering is, and then we will get into the 6 types of genetic engineering that you're going to need to know for your test. Genetic engineering is the process in which humans get involved in changing the genetics of an organism. Now, when we talked about before we discovered DNA, and then even shortly after we discovered DNA, all we knew was that chromosomes were made of DNA, and that this DNA allowed us to know what our characteristics were going to be. We did not have the technology, however, like we do now. As soon as the technology was invented, now humans have realized that we can actually go in and change the genetic code, the order of your nitrogen bases on your DNA strand. When we go in and we tamper with that DNA, that puts it into one of these next categories of genetic engineering. The 6 categories we're going to talk about gene splicing, cloning, selective breeding, gene therapy, stem cells, and GMOs. Now with each of these, we will talk about what the definition is of each of these types of genetic engineering, and I will show you examples of them. One of the very common types of genetic engineering is called gene splicing. Because all living things have DNA, and it doesn't matter if you're the smallest bacteria all the way up to the largest blue whale, all DNA is made up of the same 4 nitrogen bases: adenine, thymine, guanine, and cytosine. Now, because every living thing has these same 4 pairs, the A, the T, the C, the G, we have found that we can take little pieces of DNA from one living thing and put it into the DNA of another. When we do that, where you're mixing DNA from more than one organism together, this is called gene splicing. So if you look at the example on this slide, if we look at the top piece on the left, the yellow portion is the section of DNA that we want to take, remove from that strand of DNA, and we're going to put that into the DNA of another organism. So on the right, it will show you where that yellow strand is being removed, and then we're going to cut out the blue piece and replace it with the yellow. So we're actually taking that yellow section of DNA from one living thing and putting it into the DNA of another. Now, that new DNA strand, whatever organism that is that will go in, will now have whatever characteristic that little yellow section of DNA codes for. So here are some examples on the following slide. All of the animals on this slide have undergone gene splicing with the DNA from another animal. With this other animal, a small portion of DNA that codes for the glowing, the bioluminescence, was placed into the DNA of the pigs, the mice, and the fish. Now you'll notice up in the corner, underneath a black light, the snout and hooves of some of the pigs will glow. And in the top right picture, you'll notice 3 of the baby mice have had the gene splicing done and 3 have not. The 3 that are glowing under the black light have DNA from another animal. And on the bottom, the DNA from the glowfish. These fish, you can buy them actually at Walmart, sells them. You can actually go there, and if you keep these fish in a tank with the black light on it, they will kind of glow neon colors, and they're very pretty to look at. Now with these 3 types of animals here, with this gene splicing, this really doesn't have that scientific of a purpose. It's not like they were trying to medically show something. These are more just for show to say, hey, look what we can do. Now, I want you to think, what animal has the ability to glow that they could have taken the DNA from for these animals. If you said jellyfish, you are correct. Jellyfish have the ability, or some jellyfish I should say, have the ability of bioluminescence, which means living light. They're able to produce their own light. These are going to be your jellyfish that live a little deeper down in the ocean. But scientists have found that section of DNA that codes for this ability to glow, and they took this DNA and put it into the DNA of those animals from the previous slide. Here is another example of gene splicing. In this example, we take this DNA from a spider, and this section of DNA codes for the spider's ability to make the spider silk that it spins its web out of, and they have put this section of DNA into a goat's DNA. Now, once this goat is born and grows up, this female goat, as it produces milk, can produce the spider silk in its milk. As you see, the goats in this picture look like normal goats. However, they have been genetically engineered so that when they produce milk, they are producing the spider silk in that milk. Once the spider silk is collected and woven together, it makes a very, very strong thread. And now we're using that thread for different purposes. One example of the use for this thread is there are government agencies out there that are trying to use this thread to create a lightweight body armor for our soldiers. Something that's going to be extremely strong and protective like an armor, but also be lightweight so that they can be comfortable. The next example of genetic engineering is cloning. Cloning is a type of genetic engineering where you take the DNA from one living organism and you are going to use that DNA to make an identical copy. Now, when I say identical, the DNA will be an identical copy, so it'll be like an identical twin, but not formed like an identical twin forms. In this process, what they will do is they will take a cell from the animal they wish to clone. So in this picture, if you look at the top left, you see the sheep that is completely white. Then, from that cell, they will take out just the nucleus, and then they'll throw the rest of the cell away, because all they want is the nucleus that holds the genetic information. Then, they will take an egg cell from the second sheep in the picture, the one with the white body and the black face and the black legs. That's a female sheep. They will take an egg from that sheep, take the nucleus out of that egg, and throw that nucleus away. Then they will take the nucleus from the first sheep, put it into the egg of the female sheep. They use a little bit of stimulation on that cell to get it to start to divide through mitosis. And once the baby embryo begins to form, they put it back into the female sheep that will then carry it while it develops. And when she gives birth to the new baby sheep, it'll be an identical copy of the very sheep from that top left corner of the picture. This is the cover of a Time magazine. On this cover, you will see two sheep that look exactly the same. This is Dolly. Dolly was the first clone that was documented to the public. And she was created in, I think it was about 1996 that she was created. But they took the DNA from a sheep, just like I showed you in the previous slide, and they put it into the egg of another sheep and allowed that sheep to carry it to term. And when the baby was born, Dolly was our first clone that, again, was made public. Now, I know in your mind you're thinking, oh my gosh, how cool would it be to have a clone of myself? There are some things about clones you need to understand. The clone needs to start from being born as a baby. So if you were to clone yourself now, you would not have an identical 11 or 12-year-old clone sitting next to you. That clone would have to be carried for 9 months and be born as a newborn baby. So then they would need to grow up to reach your age. So by the time they reach your age now, you are going to be twice your age. So cloning is not going to be just an instant twin for you. There are some other disadvantages to cloning, and they were able to see this when they cloned Dolly. Dolly actually died of lung cancer. Now, they originally thought it was because she had aged quickly, and the debate that they had was, since you're starting with grown DNA, does the age of that DNA affect the health of that new clone? Another problem with cloning is it's extremely expensive. The technology needed to make cloning happen is extremely expensive, and it's not like you can run out and just go buy it and clone in your basement. The third disadvantage of cloning from this slide that I want to talk about is that if you clone an entire group, that's where the word flock comes in the slide. If you have an entire group of cloned animals, all of their DNA would be the same. So if a disease were to be introduced to this group, none of them would have any resistance. And a lot of times if you have different genetic codes, some people will carry a trait that allows them to be more resistant to a disease than someone else. But in a cloned flock, everybody in that flock would have the same DNA, so a disease introduced to that group would wipe out the entire group. The next type of genetic engineering is going to be selective breeding. Now, with selective breeding, we're not actually going in and tampering with the individual strands of DNA. With selective breeding, we're actually choosing two organisms that have desired characteristics, and we're choosing to breed them together. This picture on this slide is of a Belgian blue cattle, and you can see he's extremely large and muscular. How they selectively bred for this Belgian blue cattle was they picked the largest, most muscular cows, and they bred them together. And then after a while, if you continue to do that, you eventually end up with this very muscular cow that has a lot of meat on it for meat production. We have also chosen to use selective breeding with our chickens. If you look in this picture here, back in 1957, the average chicken was not that big and bulky. It was actually quite small. Then selective breeding began, and we started to pick the biggest chickens in the group to breed together, so that by 1978, the average now for a chicken was actually larger than it had been. And then we continued to selectively breed, picking the largest chickens to breed together so they would have the larger chicken babies. And by 2005, you can see that the average chicken was much, much larger than what it originally was in 1957. Now, we continue to do this, and you never know what the results will be. Now, I know we're not ever going to have a chicken with horns and red eyes and jet packs on its back or anything like that, but you see where I'm going with this picture. If we continue to genetically engineer the chickens, we don't know what we might end up with in the future. Another example of selective breeding, when we talk about hybridization, mixing two different types of an animal together, or two different types of a plant, or two different types of living things. Now, side note on doing this, they have to be closely enough related for reproduction to happen. You cannot take a dog and a cat and breed them together to get a dog-cat or cat-dog. That does not work. But when we look at hybridization in animals, Probably the most common to see, and most of you would know about, would be our different dog breeds. One example would be the Bull Mastiff, which you see in the center bottom picture here. The Bull Mastiff is bred from a Mastiff, which has the characteristics of big and strong, which I have bold printed in the picture. Now the problem with the Mastiff is, even though it was big and strong, it wasn't a very fast dog, and it wasn't a very aggressive dog. So if, on the other hand, if you take the Bulldog, it is a fast, aggressive dog, but the Bulldog lacks strength because of the shape of its body. So if you breed a Mastiff and a Bulldog together, you get a Bull Mastiff, which has the good characteristics of both of the beginning breeds. You have a dog that is big and strong and fast and aggressive. On this slide, I want you to start by looking at the top 3 pictures. The picture on the top left is of a Labradoodle, which is a mix between a Labrador and a Poodle. The one in the middle is a Schnoodle, which is a Schnauzer Poodle mix. And the one on the top right is an Aussie Doodle, which is an Austrian Shepherd Poodle mix. Now, when we make Poodle mixes, it is to try to get the best qualities of both breeds. Many people are allergic to dogs, but even if you're allergic to dogs, a lot of times you can own a Poodle because it does not give off the skin flakes, called dander, that cause people to be allergic to them. So it's kind of like an anti-allergy dog. But by breeding them with these other breeds, you can also get the good qualities of the other type of dog that is in the Poodle mix. Now look at the pictures on the bottom. The bottom left is called a Cheagle, which is a mixture of a Beagle and a Chihuahua. And the one on the bottom right, which is the cutest little dog, he almost looks like a little stuffed animal, this is called a Pomsky. It is a mix of a Pomeranian and a Husky. Now these two dogs at the bottom are extremely cute, and they're bred to stay small, like the Chihuahua or like the Pomeranian. Now the problem with these breeds is that there is no guarantee that they are going to stay cute. They could, I shouldn't say stay cute, they're going to stay cute, but stay small like the breed that is the smaller breed. However, sometimes when you breed dogs like this, you're taking a chance, because there is no guarantee that the Cheagle is going to stay small like a Chihuahua. It could have more Chihuahua traits, but in the size of a Beagle's body. And the same thing with the Pomsky. Even though this picture, he's cute when he is little, it could go the other way, and it could have more Pomeranian characteristics with the size of a Siberian Husky. And the problem with that is, if you're looking for a small dog because of where you live, if this dog does get big like a Husky, it might be too big for the space you're living in. Another example of selectively breeding in cattle would be the creation of the Santa Gertrudis cattle, which is a mix between Brahman cattle, which is resistant to heat. The only problem is the meat they produce is not that great. And the Brahman is mixed with an English Shorthorn cattle. Now the Shorthorn cattle, it doesn't do very good in hot climates, but its beef is very good. So by mixing these two animals together, you're hoping to create one that does have a resistance to heat, so it can live in warmer climates, and still produces meat that would be good to eat. Not all selective breeding occurs in animals. At the top, the two pictures are both bananas. The one on the left, however, was what the original banana looked like in nature. However, it wasn't very big, its flesh was not very tasty, and it was full of these large seeds. Over time, by selectively breeding the ones, choosing the plants that had less seeds and a better tasting flesh part in the center, we were able to eventually genetically engineer the banana that you are familiar with today, that you buy in stores and eat. Also, sweet corn at the bottom. Corn also has a story very similar to the bananas. When corn first started, naturally, it wasn't that sweet tasting, and it wasn't the prettiest corn on the cob to look at, but eventually, over time, with selectively breeding, we were able to come up today with these corn you see here in this picture, which is actually a very good looking, a very tasty looking piece of corn, and it does have a very sweet flavor to it. On this slide are some examples of hybridization in plants. Just like with the dogs, where we took two breeds to create a new third breed, we can also do that with some of our plants. The Tangelo and Orangelo are both crosses between either a Tangelo or an orange with a pomelo, which is very similar to a grapefruit. And it will still have the great citrus flavor, and you'll get a mixture of flavor like between the orange and the grapefruits. You'll find something like a flavor right in the middle. Your Myers lemon was created to make a sweeter lemon. So if you look at the two pictures of the lemon, the one on the left, which is has the pointier ends and is more yellow in color, that is your very sour tasting lemon. But the Myers lemon is actually made to be much sweeter, so it's used more in a lot of dessert dishes. The Loganberry is a cross between a raspberry and a blackberry, and its flavor is somewhere like a mix between the two. And in addition, the Loganberry also has been selectively bred to have less thorns on its vine, so they're easier to harvest. The Limequat is a mix between a key lime, which is used in a lot of desserts for its nice, tangy citrus flavor, along with a kumquat, which is a plant that grows in very cold weather. Currently, your key lime trees have to grow in warm weather, but it is a very delicious flavor for a dessert, so they wanted to breed it with a kumquat plant so that the limequat can actually still produce the wonderful citrus flavor for desserts, but be able to grow in colder climates. The next two types of genetic engineering I've summed up together on one slide: gene therapy and the use of stem cells. With gene therapy, this is a type of genetic engineering where you are going into DNA that is faulty, something that codes for a genetic disorder, like diabetes. You can go in, find the faulty section of DNA that's telling, for like diabetics, their pancreas not to produce the insulin they need, and then you take a healthy section of DNA from somebody who is not diabetic, and you replace that bad section of DNA, hoping to get rid of genetic disorders. Now, with gene therapy, this is more of a newer type of genetic engineering. They are working on it now. They're hoping to be successful with it, but we're not quite there yet. It's going to take some more work. The use of stem cells is a type of genetic engineering that is using cells from early on in development, which are called undifferentiated. That means that these cells don't have a final destination yet. They are not set to become an eye cell or a heart cell or a liver cell yet. They're just kind of in limbo. And what we're using stem cells for is to trigger them to become a certain type of body cell to replace faulty cells that you may have. Now, this picture actually sums them both up together by using stem cells for gene therapy. The last thing we want to talk about with genetic engineering is the term GMO. GMO means genetically modified organism, and this is any organism that has been changed in some way through genetic engineering. So on this slide, I've shown that a lot of our foods are genetically modified, and you would see that on a label somewhere on the food. Corn, soy, cottonseed, papaya, rice, rapeseed oil, potatoes, tomatoes, most of your dairy products, and peas are the top 10 most commonly modified foods that we have. This last slide is just summing up GMOs. On the top, it talks about what GMOs are and what GMOs are not. The couple sections I want you to look at, though, are starting on the right. Let's look at prevalence of GMOs. Most of what you eat has probably been genetically modified in some way. There are currently over 30,000 GMO products on grocery store shelves. If you look at the three pie charts, the first one represents soybeans, the second represents cotton, and the third represents corn. The yellow part of the pie chart is what percent of those crops currently in the U.S. are genetically modified. So you can see that there is a much larger percent genetically modified than the little green sliver, ones that are grown without any type of genetic engineering taking place. Now, at the bottom of the page, it talks a little bit about public opinion about GMOs. A very large percent of our population feels that genetically modified products should always be labeled in the store so that people know and then can choose whether or not they wish to purchase that product. And about 53% of the public says they would not buy genetically modified food. And that is for the reason that is in the bottom left-hand corner of this slide. The long-term studies of genetically modified organisms. Genetically modified food has not been around long enough in time for us to do long-term testing on. So we do not know how constant consumption or eating of genetically modified foods, how that will affect us in the future. Can it create problems with our body? That's something we don't know because they haven't been around long enough. Now, we have now finished our genetic engineering note taker. In class, we will be having further discussion about the pros, the goods, and the cons, the bads, about genetic engineering, and we will be discussing your opinion on this topic.